Okay, good morning, everybody. I think I'm ready. So many things to toggle through. Any questions or concerns before I jump back to where we left off? All good. All right. I was hoping last night, I'm like, man, snow day. Nope. All right. I know we talked about pasture and Coke, but I opened it up right here just because I'm like, I'm pretty sure this is where I left off. So, two, we will talk about repeatedly as we move through here with some of their discoveries and importance to our understanding of microbiology. And we'll go through Koch's postulates, although I already kind of went through them. We'll go through that stuff a little bit later as we speak about some disease process. So this, although we're going to think in the next chapter about some potentials that we'll think about in the lab, this kind of sets the stage for some of what we will be thinking about almost all of the time in the lab that starts next week. So dependent upon the situation, you will have a sample. Maybe it's something in a clinical situation that you have collected from a patient. There are various types of specimens that we might think about getting out of or from someone. Or it could be a lab situation where you roll in and you're provided with some test tubes full of some broth, and then you need to do something with it. Typically, what you're going to get in the lab is going to be in a little test tube with some broth in there and the bacteria. Sometimes you'll get it a little bit differently, but usually that's what's up. All right. So it truly kind of depends on what's going on. If we were thinking a clinical situation, there's a lot of things that go into truly trying to figure out what you're dealing with in the first place. And if we're thinking a bacterial infection, we will collect that specimen, and then we most likely would perform some staining techniques. We'll talk about gram staining in great detail. But some sort of staining techniques that ultimately are going to clue us in to a little bit about that bacteria. And then that opens up the avenue for thinking about, great, I've got this type of bacterium. What can I grow it on? And dependent upon where that specimen came from, maybe you've got some potential hints about what it could be of what you should be doing. All right. I know rolling into the principal's lab, you're not going to know that. I know as an undergrad, when I was going through a bunch of different microbiology classes, I'd roll in and have a handful of plates, and I'm like, I don't even know what the hell this plate is, right? It takes time. It takes practice to go through that and really get a feel for what you're working with. And we understand that. We don't expect you roll in and know what truly should be going on. That's why you're here, right? So after we stain it up, then we typically think about cultivating it, growing it, all right? So this Petri dish that we see here, plastic dish full of some sort of media, agar, and nutrients that the bacteria would like to feed on, all right? The agar is basically just a solidifying agent. They don't really eat that stuff. They eat the other ingredients that you put into the medium. Lots of different types of sugars are really what we think about. There's others where you've got handfuls of types of proteins. There's also going to be some sugars in there. Different things that microbes may want. And it really depends on who we're thinking about. Bacteria that are growing on our body, they like all sorts of nutrients. But bacteria that are growing in the environment, maybe in a water sample or out in the soil. The types of nutrients that they have as a fuel source are very different than thinking about nutrients that we could be providing. And so how we might grow them could be tremendously different, right? Things like coming from the water, typically very low nutrient levels. I think you'd hope that. Low nutrient levels for those microbes, and to take it from the water and expect it to grow up on something that's full of all sorts of nutrients, probably not really going to be very good for that bacterium. It wants something that's very light, very minimal with the nutrients, and then it will grow. All right. It also depends, thinking body and environment. Temperatures are also an important thing when we think about trying to grow bacteria. Stuff that likes to grow on our body probably will grow at room temperature as well, pretty likely. All right. But then thinking about things that would prefer room temperature, bumping that up to our body temperature, maybe they're not so happy about it. And you'll do a little of that in the lab with 2 different incubator temperatures, trying to see, do these bacteria grow at this temperature or not? Do they grow better? And sometimes you potentially see things like maybe a pigment that bacterium can produce, and maybe it only produces it at 1 temperature. Those are things that help to clue us into understanding a little bit about those microbes. All right. So this Petri dish was inoculated with some bacteria. This is called Legionella, causes Legionnaire's disease, a type of pneumonia. All right. What has happened on this plate is a technique that you're also going to learn in the lab. All right. We call this streaking for isolation. If you have a culture that is mixed, it's got a couple of different bacteria in there, you want to separate those things out. You would like to get isolation, individual colonies. Each of these, if I can get my pointer here, each of these we would call a colony. All right. So with this plate, it's really like we've done some diluting, but we're diluting it on the plate. All right. So there's multiple points on this plate where we apply the bacteria and then we spread it out. Initially, the very first time, you will take your inoculating loop that we see here, you'll heat it up in the Bunsen burner, you get it red hot, and then you pull it away from that Bunsen burner and you let it cool off. 20 seconds-ish allows this wire to cool down. All right. If you touch your bacteria, whether it's in a broth or on the Petri dish, and you hear it sizzle, that's bad news. All right. Sizzle means death. All right. You don't want to kill those bacteria. You want them to live. All right. If you hear it sizzle, it's not the end of the world. Just take that loop, heat it back up, cool it off a little bit longer. Now go 30 seconds and don't blow on it. Don't wave it around in the air. We've tried to sterilize it and keep anything that was on it off of it, right? So you just hold it there, kind of close to your fire, but not in it, right? As it cools, then you dip it back in the broth or you touch it onto the Petri dish. No sizzle. We're good to go. All right. I take a loop full of that bacteria and I swab it all over in this first little bit, this quadrant, if you will, if you could think of it as breaking it into 4s. Sometimes we do a T streak and it's 3rds. Doesn't matter. How you do it, I don't care. As long as you get some of this stuff, you're good to go. All right. So you swab it on there with your loop back and forth, and the loop just gently glides right over the surface of the agar. All right. We're not farmers, although we are cultivating. All right. We are not farmers here. We're not plowing into this dish to try to really get it in there so it can root in and grow. They don't do that. All right. They just glide right over the surface, and those bacteria will start feeding on the nutrients we've provided. All right. So after my first swabbing onto this plate, that's the only time I'm adding bacteria. I take my loop, I put it back in the flame, I cool it, I let it sit for 30 seconds, and then I go back to where I had done my initial swab, and I dip into it a couple of times. This looks like quite a few times, all right? A handful of times going into it to grab a little bit of what we laid down initially and spread it out. I flame it, I cool it, I do it again. A couple of times, all right? This looks like 1, 2, 3, 4, 5 different times, all right? 3 times, 4 times, 5 times, doesn't matter as long as you get some isolation. So each time, in between the spreading and the re-sterilizing of that loop, when you go back into it, you're grabbing less and you're grabbing less. It's a dilution, all right, on the same plate. So as we get to the 3rd or 4th or 5th in this case, even though the 4th, there's some individual colonies, all right, you start seeing separation. Instead of a swath of growth that you probably can't really tell, is there more than one thing growing here? Because they're all slammed together. But when you get them separated, isolated, now you've got the hint of knowing this is only one type of thing, likely because they all look the same, or potentially a couple of different things because the colonies, the shape, the characteristics, colors, all sorts of things, as you make these observations, clue you into same or potentially some different ones. We typically like, especially if we're thinking clinical situation, to get a pure culture. So if what we have is a mixture and we streak it out and then we see those distinct colonies, we want to pick each of those and put them onto separate plates and grow them up. So the next round, as we observe it, all the same, instead of 2 different things on the same plate. When you verify that's all the same stuff, now we're saying you've got a pure culture. Now it's this one thing, all right? So all sorts of different media. I'm not going to bog you down with details about that right now, but they help us to separate, to distinguish, and some just flat out help us to cultivate, just gives them stuff so they can grow. But others can clue us in. And this is a specialized plate that allows Legionella to grow. Many bacteria, as I've already kind of hinted, want some specific things. And if you know that, then you've got a greater likelihood of actually getting, all right? So you will do this stuff a lot, all right? So you'll roll in there and you're like, I already know how to streak for isolation. I learned it, right? Doing it is what truly needs to happen, all right? So we got to talk a little bit about Linnaeus. I feel like it doesn't matter what biology you're in, Linnaeus has to be thrown up in there, not throwing up in there. So he was a botanist, zoologist, and a physician, and ultimately, I think, you know, in biology, there are so many names, and we think of all of the scientific names that can be mind-boggling to try to wrap your head around. Deep down, we can thank Linnaeus for that. He's the one that set this process in motion. There was already some thoughts about trying to categorize stuff, and it was super simple. And a lot of it, really, we could think Linnaeus setting up some of this. You've got all sorts of living things. How do you know? What are we really talking about? And so he thought we should categorize them a little bit. If you move, you're an animal. And if you don't, you must be a plant. Done deal. And we start grouping that way. All right. Technically, as he was setting all of this stuff up, there was another group in there as well. He had some minerals thrown into the mix, which technically we could think some of that stuff essential for life, but it's not alive. That's not on this classification. It's been removed because we're like, wait a minute, that really doesn't need to be here. All right. If you want to understand that stuff, take some geology, right? Then you'll get that, right? Go through that mechanism. So all sorts of ideas, truly starting from Linnaeus to get this bit of separation. But I think all of us understand plants and animals isn't sufficient, right? We need some more. We need some more ideas, all right? I like to think of this like your house or your parents' house. Probably in the kitchen, there is a drawer that you call the junk drawer, right? That's where you get all sorts of crap that collects there, right? Likely, there is some point that junk drawer gets full. And at some point, you're like, crap, I got to clean this sucker out, right? And you try to do some separating, some sorting, rearranging the littlest bit. That's kind of what I think about with this classification scheme. Too much stuff. Let's get some more drawers up in here, right? Let's get this filing cabinet so we can start categorizing it a little bit better. Linnaeus started it, and many others following through as we get better understandings about all sorts of diversity of life, start adding different drawers to this understanding. All right, we'll hit that right now. Haeckel shifted it up. All right, and we can see this tree of life, if you will, and we've got our plants and we've got our animals, and then he's like, there's some weird stuff that doesn't really fit either of these categories. They're microscopic. They typically move around, but not all of them. What the heck is this? These are the protists, all right? So he gives another branch into this, protista, all right? And as we move through there, we're going to divide it a bit more. We'll come back to the protists because right now, they're our biggest junk drawer. There's a lot of stuff going on with them that we're like, wait a minute, we ought to separate this out a little bit better. All right. We're getting there. So Linnaeus and minerals would be on here, right? Animals and plants is our first bit. Haeckel separates it out with the protists. And a little bit later, he's like, wait a minute. Here's some other crazy microscopic stuff that doesn't seem like the protists, right? The monerans are our next players to get added to this tree of life. And these guys, they're the bacteria, all right? They're also the archaea. We'll separate them out here in a bit, all right? But monerans are these microscopic, what we will define as prokaryotes, organisms that don't have a nucleus or any membrane-bound organelles, super simple players, all right? Those are the monerans, all right? Even though I say simple, they're super complex, all right? So they get added to the mix, all right? So 1750s, century later, further separation, about another century, 1960s, late 1960s, Whitaker decides these fungi, these mushrooms you see at the supermarket right by all of the produce aren't plants, right? They are very different. Cell wall characteristics, all sorts of different characteristics in the fungi that are very different than animals and very different than plants, all right? We need a new drawer, all right? The fungi get added up into the mix, and now we start truly thinking the 5 kingdoms of life, all right? Plants and animals, fungi, the protists, and our prokaryotic organisms, the bacteria, or the monerans, all right? So little bit later, not another century, little bit later, handful of years later, all of the sudden, we've got the ability to examine DNA. We've got the ability now to look at genetic signatures and think a little bit more differently about this tree of life, about how things are related in the first place. And that opens up the potential to reevaluate these drawers and say, wait a minute here, right? Just because you look similar doesn't mean you necessarily are, right? So the Monarans, they get their next separation, all right? This was mid 1970s. Wose and Fox evaluating some cellular material that is shared by all walks of life. And when you want to think about understanding evolutionary relatedness, it's important to think about something that's a common denominator, something that they all have. And then you start looking at how is it different, all right? So everything that's alive builds proteins. We all need them. We build them, all right? And the protein machinery is shared by everything, all right? Ribosomes are what we're thinking about, all right? The workbench of our protein synthesis. So they start looking at ribosomal RNA, the small subunit, two different subunits sandwich around that messenger RNA, and we start building protein, right? Building amino acids. So the small subunits, there's variation between the Monarans and the eukaryotes, all of the nucleated cells. There's variation with that. The ribosomes are similar in function, but eukaryotic cells a little bit bigger, right? And the genetic signature, the profile for that, there are subtle differences in the amino acid signature that you can recognize between the Monarans and the eukaryotes. Makes sense. They're separated, and the ribosomes are different, all right? But the bacteria and archaea, their ribosomes are the same size. It's a similar idea. And these archaea were what we classified as extreme bacteria, all right? These were the super oddball bacteria that grow in areas that you flat out say nothing could live there. Archaea probably do, all right? There are some bacteria that also have some extreme tolerances, but the archaea, they can do it better, all right? So they start looking at these Monarans. They start looking at all of this bacteria, extreme bacteria and run-of-the-mill bacteria, regular bacteria, if you will, all right? And they start looking at the small subunit, the 16S ribosome, and looking at that genetic signature, right? And recognize differences between these two groups and proposed a new filing cabinet, if you will. We ought to separate these guys out, all right? So they got bumped up above the kingdom level to the domain level, all right? So 3 domains of life: bacteria, archaea, and the eukaryotes. And technically, all of those 5 kingdoms are still represented. But when we look at those Monarans, they get bumped up, all right? There's also potentials in some of this where they talk about super kingdoms and things like that. We're not going to get bogged down with that. The domains, up a notch from the 5 kingdoms, and it provides the opportunity to separate our bacteria from the archaea, all right? So as we look at this phylogenetic tree, we're rooted down here, our black bar, common ancestor. We call it LUCA, last universal common ancestor. And there's all sorts of thoughts about that, not just one single type of organism, multiple things playing into the mix that truly give rise to the diversity that we think about, and all sorts of potentials for genetic exchange that we'll touch on in a little bit later as we think about some of the potentials for these cells to interact, all right? So we start off and we see this branching point. Each of these branches on our tree we would call nodes, and a node is a speciation event where we see separations. So at this point, up from our root down here, we see a separation. Bacteria start developing, and shortly after that, maybe not so shortly, all right, we start accumulating differences, and those differences give us another node, another branching point. And you can see the archaea branching off, going up toward the eukaryotes. They harbor some characteristics that are similar to eukaryotes, and they have some that are similar to our bacteria. They're in between, and a step in between bacteria and eukaryotic cells, all right? And then we move forward from this node, and all of this kind of tannish, this is the eukaryotic lineage. Nucleated cells, organelles, all sorts of diversity, many, if I can get my pointer back, many of these single-celled. Then multicellularity shows up on the mix, and our last three on the branch show up: plants, animals, and fungi. This is us, just like the fire escape, little star on there. You're right here, all right? There's the exit. All right. I think if you go out here, you just right into a snowdrift. I don't know. Somebody went out there yesterday. Who went out? They're not even here. They're stuck in the snowdrift, all right? I don't know where that door goes, but it doesn't go, I think, where you want it to go. All right. They figured it out. And then once it closes, they're like, I can't get back in. Sorry, I hear you knocking. You can't come in. All right. This image I borrowed the text, and we will see the one that the book actually utilizes. I like this one a little bit better. All right. So when we think about microbiology, it's very hard to wrap our minds around some of what we're really trying to think about in the first place. It's like astronomy and thinking about how far away all of the distant galaxies are and all of, whatever. When you look through a telescope, you get a better understanding than just staring up to the sky. Looking through a microscope, same but different, gives us a better understanding about this invisible world all around us, all right? So I like this one because of some of the examples provided. We'll touch on the other one in a bit down the road. So this just puts some things into perspective as far as what we might be able to see with the naked eye, maybe with a lens, a magnifying glass, some detective work, all right? And then the invisible world, all right? As we get down past our micron or our millimeter range, all right, we start thinking about our ability to actually visualize these things, the resolution of it. We can't see things like a human egg or a paramecium, although there are some potentials with some of these cells to technically be quite large, and maybe with a magnifier, not a microscope, you might be able, all right? Little bit of wiggle room, all right? But this brings us in to the microscopy realm when we think about trying to visualize these cells. Down in yellow, this is what we think about in the labs that we will be dealing with, conventional light microscope. They've got great magnification and allow us to see cells, but when it comes to the bacteria, they are super small. And when you look at them under the highest power on the microscope, they're still going to look very small, like little teeny dots or little rod-shaped, as we see kind of our mitochondria and bacteria, elongated, all right? Very small, all right? Most bacteria are in the realm of about 1 micron in width, all right? Sometimes maybe a little bit longer. If we're thinking some of these rod-shaped, they might be 2 microns long, 1 micron wide. Lots of the spherical bacteria, about 1 micron, all right? Very small, all right? And in order to see that, you've got to magnify it, and it's not just a hand lens, all right? You have to use multiple lenses to get the magnification able up enough to visualize them. And most of the time when it comes to bacteria, you're going to have to put some stain on there, because if you don't stain them and you don't really know what you're dealing with, you're going to be hard pressed to see anything, right? They have, most all of them have a cell wall that you might be able to see, the little hint of an outline. I like to call those ghost cells, because you can sort of see it, but you don't see anything inside. It's just the faint little outline, all right? That doesn't do you much good, all right? Putting some stain on it lets them pop out, all right? And there's a lot of different stains that help us to differentiate some of that, all right? So light microscopy, here we can see different lenses. The oculars, and we'll talk microscopes in the next chapter, they magnify 10 times. And then the objectives, the lenses close to the slide, go from 5 up to 100. So you multiply all of these by 10. The highest magnification is 1,000 times, right? We are bumping up these 1,000 times their size, and they still look like a speck of dust about as you look through there, all right? Very little. To be able to perceive things even smaller, stuff like viruses, you can't see a virus with a regular light microscope. They're too small. They typically are about 100 nanometers, all right? Much, much smaller than bacteria, all right? Viruses are very, very tiny. To a virus, a bacterium is a monster, all right? To a bacterium, a human cell is a monster, all right? About 10 times difference as we think about each of these sizes, all right? Very small. Now, when it comes to electron microscopy, there's multiple steps, procedures needed to prep those samples, just like you would do with the bacteria, but some different types of staining techniques to coat it, to allow it to be visualized. And with an electron microscope, it's a beam of electrons that's shot down at the sample. And as it hits that stain, which is typically some sort of heavy metal like uranium, it refracts that, and you get the image of what was coated behind. All right, pretty impressive. There are lots of images of our bacteria that they use electron microscopy, because that gives you really good size image, little bit of detail about the surface of the bacteria compared to just a regular conventional microscope stain, all right? Although you'll see some of those as well, all right? You can get other stuff, potentially some proteins visualized with electron microscopy, but when we get into this much smaller realm, 0.1 nanometers or less, as we think about trying to visualize an atom, you can't just look at it with an electron microscope. Although there are new advances, and I believe, I should do a little searching, I believe there is an image, not of a single atom, but of hydrogen gas, H2, two atoms together. A very small molecule. I think they have imaged that, but not an individual atom. You don't see the nucleus of that atom. You don't see electrons. We're not at that point to actually visualize that, right? Not yet. All right. So a lot of variation, all right? And this is where we're truly thinking very small, all right? As we bump it up and think about like some of the protists, they're much larger. And when we think about looking through the microscope, probably not going to use the highest magnification to look at eukaryotic cells. If you do, you probably don't see the whole cell. You probably only see part of it. And maybe that's what you want. Maybe you would like to examine some of the organelles, the nucleus or something like that. Then maybe you bump it up, but typically a little lower on that. And then you can see the whole thing, but you wouldn't see a bacteria very well. So when we visualize them, and this is all electron microscopy that we've got going on down below, bacteria come in all sorts of different shapes, sizes and shapes, but size-wise, I already said pretty similar, as we're down by about a micron, all right? Although it does look like some variation, all right? You can see some of our ruler bars over here to give you a size perspective, all right? So right here, 200 micrometers is this ruler bar, all right? So our lots of bacteria are spherical, coccus, singular, cocci, plural. If you're looking at it and see boatloads of these spheres all over, cocci, staphylococci, streptococci, these are spherical bacteria. The way they're arranged, that's a slide down the road, all right? We can distinguish some of that. Here we see two of them together. And maybe we see lots of them that are doubled up like this. We'd classify that as diplococci, two spheres linked together, all right? Other bacteria, and these two right here are the most common stuff that we think about, typically what we'll be visualizing, all right? Bacillus are these cylindrical shaped cells, all right? We call them bacillus or rod-shaped bacteria. There's also a genus called Bacillus. Guess what? They look like this, all right? Bacillus shaped, all right? So rod-shaped or bacillus shaped. If that rod is a little bit different, kind of curved, kind of wider at one end than the other, some say comma shaped, sort of, all right? Sort of like a comma. Kind of looks like an eyebrow to me, just the way it's right there, right? But not a monobrow, right? Single, right? This is Vibrio, curve shaped, right? Coccobacillus, this I struggle with. These are oddballs. They are a little bit of both. Spherical, but a little more elongated. Not curved like Vibrio, but not big enough to really say bacillus and a little too oval to just say coccus. So we're like, you know what? This is coccobacillus. It's a bit of both, all right? So little kind of stubby rods are the coccobacillus, all right? A little more oval shaped. Then you get to some oddballs, all right? Springy bacteria, spirillum and spirochetes, curved but elongated like a spring, all right? And this allows the bacteria, some bacteria have the potential to move, some do not. Our last two corkscrew or spiral through their environment as they move around, and it has to do with how the flagella is kind of glued to the side of the bacterium, allowing it to corkscrew through, all right? The bacillus potentials for motility, but when we get to the coccus, we don't think of these as being motile. They don't have flagella, they don't move around, maybe gliding motility, slime layer sort of stuff, but not actually propelling themselves through an environment. We'll touch on that stuff a little bit later. Oh man, right now as I see this, I'm like, wouldn't it be nice to go to the hot pools? I wouldn't want to soak in this at Yellowstone. Stay on the boardwalk, people. I keep seeing all sorts of things in the news feeds about, you know, dummies going to Yellowstone that are like, oh, let's go feel how warm that truly is, right? Or could I ride a bison, right? You should not, right? I recommend not. Or maybe you should try, right? Weed yourself out of the gene pool, right? Maybe that's a good idea. Probably not. All right. So Yellowstone is almost right in our backyard, right? I would hope that you have had an opportunity to go to Yellowstone and see some of the features that it truly holds. You probably need to go more than once to get it all right? It is massive. So the features that we see in Yellowstone and numerous other areas, there are some areas right here in Idaho that have some thermal features that kind of bear some similarity to this. Handful of years back, we went to a hot springs just outside of Lava Hot Springs, called Maple Grove Hot Springs. If you've ever been there, awesome. If you haven't, you ought to check it out. They have some thermal features right around the actual hot pools that look super similar to this, kind of Yellowstone-like. No boardwalks, barbed wire fence, walling it off. Did I go through the fence? Yes, I did. I wanted to see it a little bit better, right? I felt like there, I probably could. I'm not getting off the boardwalk at Yellowstone, all right? But that, I was like, I think I could go a little closer. I'm not going to touch it. I'm not going to see how warm it truly is, right? But it was impressive, all right? And it almost was like this, right? So this is Morning Glory Pool in Yellowstone, and we can see a lot of different things going on here, but a lot of the color differences are the results of different types of microbes feeding on stuff within this water, utilizing minerals as a source of energy or a final electron acceptor. There are all sorts of different potentials, some utilizing sulfur and creating hydrogen sulfide, giving you kind of a rotten egg sort of odor, others using other things, all right? So some of these color differences are really the result of microbes. There's other areas, and I should have that image right up here too, in Yellowstone and other places where you can see big mounds that look kind of like rocks, but these microbial mats are the result of thousands of years of bacteria growing and growing and growing on top of those that came before them to create these features. Crazy, right? Super cool, all right? I'm not going to go through great detail right now, but when we talk some biotechnology and DNA, we'll want to think back to this, right? Our ability to replicate DNA through PCR reactions is the result of Yellowstone. There were scientists have been studying Yellowstone since Yellowstone became a park, truly. After some of the discoveries, the ability for scientists to study at will doesn't happen, right? You can still do stuff, but now it's kind of like a lottery. And if you want to do research in Yellowstone, you have to put in and hope that you get picked. I worked with a professor, this was many years back. He went to Yellowstone all of the time, didn't collect any water because he couldn't, but he wanted to. He had, for over a decade, his name was on the list and never picked. He went with some other people whose names were picked, some of these, I think University of Montana. And he had the ability to go and do stuff, but he couldn't bring any of that back to his lab. Not his deal, right? So we discovered an organism that thrives in this habitat, and another researcher discovered the enzymes that it utilizes is very heat tolerant, and that opened up the avenue for thinking about PCR reactions. How can we replicate DNA in a test tube? They figured it out. Massive amount of money, all right? PCR reactions are big business. Carrie Mullis that discovered these enzymes and the process of going through it, got the Nobel Prize, retired, spends his days surfing in Southern California, as you should, right? And potentially will lecture. We thought, this was many years back when I was a graduate student, we thought, wouldn't that be cool to bring Carrie Mullis? He could do a seminar. He could talk, more than just microbiology people would like to hear about his stuff. Everybody thinks about DNA and replicating and whatever. So kind of like a rock star is what I would say. All right. And I feel like scientists should be elevated to those platforms, right? Rock stars. We should have trading cards, right? Mine would not be traded, right? But some, you'd want it like, Harry Potter and getting the cards, right? Same idea, right? So we thought we could bring him here, right? So here's the rock star thing. Like when they say my people contacted his people, that's what happened, right? We didn't get to talk to Carrie Mullis. He doesn't talk to people, not like that, right? We talked to someone else, right? And in turn, he was more than happy to come here and talk for 50 grand. And I was like, we only have $100, right? That's what we pay, right? That's maybe shifted a little bit, but typically bringing a speaker in, you give them $100 honorarium. They talk for an hour, they make $100, right? I'd do that, right? That's okay. You also, in an attempt to get them here, you pay for their airfare, you pay for their hotel, you take them out to dinner. This is ISU, so you don't buy them alcoholic beverages on ISU's dime, right? But you can, right? As long as it's out of your own pocket, right? And then you ship them home with $100 in their pocket. So not a bad deal, truly, right? He wanted all of that plus 50 grand. And we're like, Microbiology Club has a very small budget. We don't have 50 grand for sure. And if we were to bring you here for the rest of eternity, we could never bring anybody, right? We're broke, right? So we just said thank you, right? To his people, right? And there you go. That's my Carrie Mullis story. Every time a PCR reaction happens, which I can't even tell you how many PCR reactions happen in a single day. The microbiology department, the core facility on the 4th floor, busts out PCR reactions every single day. And there are many professors who have PCR reactions in their lab. Every single one, he receives royalties, right? And you cite him in any paper because he's the one that did it, right? Nuts. He probably gets a penny, right, from every PCR reaction. But if you're doing 1 million every single day, right, that's a lot of pennies, right? I think surfing in California would be nice. All right, I will leave you with that.